Yeah, do you have that
I do.
option?
I think I do. We just started a recording, so
Good.
everybody on the call, we are being recorded. Not sure how that will all work, but we're recorded.
Great, thank you.
I'm here.
Hey, Katie.
Hi, everyone. I'm double booked, so I'm trying to be with you all at the same time.
Oh, great. Thank you, Katie.
It's good to see you all. Hi, Howard.
I just asked Dwight if he could record this, so I was hoping that we'd have that in case you couldn't make it. Hello, Michael.
Hey, Michael.
Hello. Sorry, I'm in my car, so.
Glad you could make it.
Yeah. Hey, there's Mr. Howard.
All right. Well, we got Michael. I was hoping Cassie could make it, but haven't seen her yet. But we're up to 17 participants, so we're going to get started. So again, thank you for attending this. We do have a lot of new people on this call. A lot of interest was shown by EPA, so I know there's a lot of EPA members here. We're not going to do introductions, but you know, thanks for spending your Friday afternoon with us learning about mushrooms. So
couldn't imagine a better group to spend it with.
Yeah. Yeah, there's worse things to talk about on a Friday afternoon.
Yeah, I'm happy to introduce.
Before I turn this over to Howard, I wanted to give a little bit of background on how we got to this point. So as most of you know, the Port and Metro have been working to clean up Willamette Cove for a number of years now. And we're working under a voluntary cleanup agreement with Oregon Department of Environmental Quality. So at this site, there's been numerous investigations and studies conducted over the years. And a while back, we got to the point in the process where we were asked to develop a feasibility study to look at the various technologies and alternatives for cleaning up this site. So as part of that feasibility study, we looked at some 30 different types of technologies for cleaning up the site, including bioremediation. And, you know, to be honest, when we did the feasibility study, we screened out bioremediation, and it was mostly due to, you know, the research that we had seen says this type of technology is effective, you know, primarily only for organic contaminants like petroleum products. And we just didn't feel it would address the wide variety of contaminants that we have at Willamette Cove. So again, we screened it out at that point. So fast forward to August of 2019, and Howard Sprouse was invited to present at the Portland Harbor Community Advisory Group. And Howard talked about this integrated approach of micro-remediation using various strains of mushrooms or fungal isolates, coupled with phytoremediation using plants such as hybrid populars inoculated with endophytes. So, you know, really not to understate it, the community was really interested in this technology. And so a lot of those community members at this CAG meeting looked at me, looked at Katie Wild and say, you know, ask, can we do this at Willamette Cove? So Katie and I, Metro and the Port, committed to follow up with Howard, and that's how we got to this bench test. So that's a little bit of the background. So where are we at right now? In July, we tasked our consultant Apex to put together a work plan on how we would do this bench test. They worked in collaboration with Howard at the Remediators and John Freeman at Intrinsic Environmental to come up with what we had planned to do for this testing. Apex collected about 20 gallons of soil from the site, targeting, some specific locations where we hope to get the highest concentration of contaminants that represented the site. And that's really the soil that Howard is starting from. So this is actual soil from Willamette Cove. Yeah, I wanted to also add, when we collected this soil, we had it homogenized at the site, and we sent samples for analysis to our laboratory to establish our baseline concentrations of what that soil represented as far as contamination. Also, as part of that analysis, we had some extracts drawn from the lab and sent to Dr. Robin Tagway at Oregon State University to look at, part of her studies are doing zebrafish bioassays. So some of that work was done pre-bench test, and we hope to do the same once the bench test is complete. And I'm not going to get into the details of that, but we're hoping, one of our next virtual tours, we can coordinate with Robin or her team and do a tour of her laboratory and, learn more about her studies. So I wanted to add that. So, finally, we expect this tour that Howard's going to take us on here about 30 minutes. I think that's right, Howard, is that right?
Yeah, 30 minutes. We'll cover what we can. So
we're hoping we have plenty of time for questions at the ends. So maybe to help with, just the logistics and the noise, if everybody could mute themselves while Howard does his tour. We'll again save questions for the end. If you have questions that you want to that come up as part of, as we go along here, you can add them to chat or just hold them till the end and we'll address them then. So with that, I'm going to turn it over to Howard. Thanks.
Yeah, thank you, Dwight, and thank you everyone else here. I'm going to expand just a little bit on what Dwight said, and I think I'll start with talking about the association, how we got here. This is a very unusual and I'd say privileged opportunity, and thank all of you. I know a lot of the people that are listening here have taken part in being responsible for this, and with that I met the Community Action Group through the Northwest Toxic Cities Coalition, which has had a meeting every now and then over the past decade, where they have assembled teams of people that have different pieces of human health and environmental health and safety expertise in that. And this last year, we met groups in Seattle, which I think was probably one of the best organized meetings that we've had so far over the last decade. There was somebody there that had expertise in the effects of lead on developing brain chemistry, for instance. There were people there like myself who had pieces within the remediation industry. There were healthcare professionals there, and there were people who, a person who represented integrated solid waste processing technology. And anyway, that's how I met the Community Action Group and how I got here. Well, that particular meeting happened on campus at the University of Washington. And over the last, I think it's probably been 12 years now, when I first met Dr. Sharon Doty. She runs the forestry lab at the University of Washington. She's been focusing on the integration, exploration, and development of the use of hyperaccumulating plants and microbes that have the capacity to either transform or remove toxins from the environment. And Dr. Doty and I had worked together on and off over the years, mostly trying to find places to do a concept in the ground trials and so forth. And so anyway, Dr. Doty introduced me eventually to John Freeman, who's here. And John works for Intrinsics Technologies out of Building 19 down on Moffett Field at the NASA Ames Research Park. He and his colleagues are really at the tip of the spear in the development and actual use of phytoremediation that combines microbes and plants for the purposes of treating, cleaning soil and water. And so Intrinsics and the remediators has been working together on and off over the past several years. We've had a number of projects. We had very successful one down in Central Valley of California that is at an old fuel depot that had contaminated the local watershed. What I'm calling the integrated biological approach, which is the synthesis between phytoremediation and microremediation technology, was employed down there, and it allowed the client to remove their very expensive and not completely effective technology that was used for cleanup. And in a nutshell, the way that the system works is that we're able to deploy the use of cuttings of hybrid poplars that are usually selected specifically for their ability to both live within the environment that we're going to be deploying them in, as well as having the capacity to hyper accumulate toxins. And what that means is that they are able, against a reference amongst these types of plants, to uptake contaminants at a higher rate, basically. And so that, combined with a suite of microbes that we've deployed here in this test, allows us now, and really for the first time ever, to treat mixed organic and inorganic contaminants, halogenated compounds. These are compounds that have chlorine, fluorine, bromine, all of these halogens in them that formerly couldn't be treated using independently any of these technologies. And so this integrated approach that we're deploying here, we're trying to demonstrate it specifically on dioxins and furans that are in the Willamette Cove soils. But these soils also contain a suite of other organic and inorganic contaminants. And this, for reference, we have materials online about this, and you can all view how the trial was set up and sort of the technical details around that. And I won't go into that too much today. But I want to say that this is special really for a number of reasons. And one is that generally when you're testing remediation technology, you usually don't get to do it with the actual soils from the environment that contain the toxins. And so you miss this component of having hopefully similar biological, ecological attributes that we're trying to mimic in the laboratory here. And I know whenever you dig something up and move it, this is affecting it. But this is something that differs from most research in remediation in that in most of the academic research that's been done, we use compounds that are then put at a specific ratio into an engineered soil and so forth. And so it really has little comparison with the actual environment that it came from. And so that's one thing that you don't often get the chance to do. And then the other thing is that there are a lot of restrictions against even bringing stuff like this into laboratories. This is something very hard to do in a lot of colleges. We're very, very fortunate here that we've been allowed to work with eco-analysts who I'm housed in their facility. They do marine toxicology, mostly saltwater toxicology, a lot of work on sediments. They do very similar work to what Dr. Robin Tanguay is doing down at Oregon State, only this is on the marine side, the saltwater chemistry. And so with that, thank you for joining me. Welcome to my home. And I'm going to show you a little bit here in the lab. I and the owners of this facility built this laboratory here specifically for doing microremediation trials. Oh gosh, I think it's probably been 15 years ago, maybe longer at this point. But anyway, I come and go around here. I've had a presence at the lab, usually helping them to build their machinery infrastructure and stuff like that. But anyway, I'm home again. And so I'll show you around the room here. First of all, I'll walk right on over to, this is a sterile hood. And what that does when we turn it on, listen, you get to hear it run. And you won't hear me as well, but this takes in ambient air in the room, it blows it through a superfine filter, and it creates a presumably clean, sterile environment to do our work. And doing sterile culture work, I wish that you'd been here a little earlier. I'm going to walk you right over to this and show you what I've been doing today. Here, excuse me. Here in these Petri dishes, can you see them? I'm on the wrong side of the door, so let me know if you can see this stuff.
We can see it.
Okay.
Yeah, we can see them.
Yeah, here what I've done today is over in 3 of these Petri dishes, I found a culture growing not far from my home of a type of fungi that has antibiotic properties. Anyway, it's called Leucobacillus albismus. And I've been wanting to do a tissue culture of it for a number of years. And so we're trying that out. And also in the center, here's a Petri dish. I like to find out whether or not my technology is working well. And so that Petri dish has agar in it. That is a nutrient-rich media that you can grow either bacteria or fungi on. And I had it sitting in front of this hood for about a half hour uncovered to see if any bacteria showed up. And so I'll be checking this in a few days. And if there's no bacterial growth on there, this thing is golden. So anyway, we're coming over here to our trial and I'll show you exactly what's going on here. Now, we have 3 separate strains of fungi that we're using in this trial, along with the poplars and a suite of endophytes that Intrinsics Technologies has supplied us. And they've been mixed in with the soil from Willamette Cove at a ratio for the fungal treatment at 20% ratio against the volume of soil. And we have 3 containers each for each strain. And I'm using a couple of them that we've had in our strain library. One is called Trametes versicolor. It is a mushroom that's also kind of a popular medicinal. It grows on dead softwood and hardwood trees. And then I'm growing a type of oyster in here, and I normally don't use that, but this particular one thrives really well in soil, unlike most oyster mushrooms. And it also has been a known degrader of hard to treat compounds, including halogens. And then the third one is one that I call the lab rat of fungal strains. It's called Phanerochaete chrysosporum. And it's really a good reference because of it's been broadly tested. And so we have a really a good spectrum of organisms to work with here. And so what we're hoping is that by the end of this trial, we're going to be able to see a difference in response between these, and this can be used as part of the selection process for the fungal strains that we're using. And now these poplars are a special variety of hybrid poplar that has a tolerance to the type of soils that are down in Willamette Cove, and also a tolerance to selenium. And there's a suite of 3 separate microbes in each of these that have been known degraders of similar compounds to the ones that we're working with right now. And so, you know, you don't see a lot going on in mycoremediation normally because most of it goes on underneath the soil. But the thing about phytoremediation, you see a response really right away in terms of the tolerance and the vigor that these plants take when they grow. And if you notice here, we have 2 cuttings per container. Now, the taller ones had roots on them, and John, you were right, the roots are rather fragile. And so anyway, we also planted unrooted cuttings, which are the shorter ones in here, and they are taking root and they're growing really pretty doggone good here. And then if you notice over here, I hope you can see the white at the surface. That's an overgrowth of one of the fungi that it seems to be tolerating the soil real well. And so anyway, we're feeding these things just water, deionized water from the lab. We're testing the moisture and trying to maintain it at a decent and not overly wet and not dry condition. And then down below we have controls set up. The 3 down here are just controls of the soil itself with no additives. It's just getting watered. And then the rest of them are a soil from a nursery, and we're just looking for a growth response in there. And so anyway, this is where I spend a lot of time, and usually with the hood going, and this room is a semi-sterile environment, and so I don't have to wear a mask when I'm in here, and I'm kind of isolated from the rest of the lab. What I want to do is I'm just going to give you a little peek outside the door. I talked with the lab managers today. We're not going to get a complete tour, but I'll just give you a shot of some of the stuff that's going on here. As I said, it's a marine toxicology lab. The kind of work that we do here is a lot of studies of toxic sediments, for instance. We work for municipal clients, a lot of oil company work and so forth. And all of it is pretty private. And so the lab manager didn't want me to be photographing everything. But I'll give you a little peek. This is really, it's one of the premier facilities in the Northwest for this kind of work. See, what we're looking at here is we have water that is piped in from out in Puget Sound, and it goes to these areas where we can put tanks and aquaria and do all kinds of studies here. There's some stuff going on.
Howard, we're having problems seeing.
Oh, there. Thanks, John. Yeah, I think I had my finger over the camera. Can you see now?
Yes.
Yeah. And here we have some mussels that mussels are a real good indicator of effects, toxic effects from the water that they reside in. They tend to accumulate these toxins inside their tissue. So the what it takes to get to this point, I think that we'll be able to do this at one of our next meetings. I'll show you how we actually set up the trials and then the theory behind them. And you know, basically what we're doing is we're mixing the in this case the fungal mycelium that we've grown for the trial, and here's some that's left over that we didn't need to use. This is semi-sterile as it grows, and so this is called spawn, and the stages that it takes to go to this, and then eventually to the point where you can actually use it for remediation are: you start with a mushroom, you end up culturing that mushroom in petri dishes. Then you end up taking those and expanding the amount of that culture, ramping it up, usually by an order of 10 each time. And so one petri dish becomes 10 petri dishes. That ends up being blendered and used as a liquid inoculation to either grain spawn or sawdust spawn. This stuff here happens to be sawdust because the type of media that we normally use in a remediation is a cellulosic carrier, something like alder sawdust or alder chips. And the reason that we use that is that if we think about the degradation of rotting wood, and then compare that on the biochemical level to petroleum hydrocarbons, it looks really similar. That wood is a combination of polymers, lignin and cellulose, and as it breaks down, the breakdown compounds are very, very similar to what you see when you're breaking down petroleum itself. And so when you grow mushroom mycelium on wood, the wood becomes a carrier of these organisms, as well as providing a food source to these organisms as they're then able to degrade the toxins around them. And the unique thing about the way that fungi work is that, unlike people, we have stomachs. If you're a mushroom, you don't. Everything that you do happens outside the cell wall. It happens out in the soil environment. And so they excrete enzymes. And then these enzymes break down compounds that the mushrooms then reabsorb and transform and grow through. And in that transformation and growth, they break compounds down. And so in a similar way, the microbes that are present in these plants, they also have the capacity to do the same thing. And so something that there's a synergy that happens when you use both of them together. We try to select fungal strains that play well with the suite of microbes and the plants that they're growing in association with. And the relationship is, I think, most clearly stated as one where the trade-off is as these plants grow, they produce sugars and they produce an energy source that then the fungi can live on. And in exchange, the fungi are able to break down compounds that are combinations of organic and inorganic compounds and put them into a soluble form, whether or not they are able to grow on them themselves, but into a form that the poplars can then uptake into them, and at the same time that the microbes that are contained within the poplars and in the soil surrounding them are able to degrade. That about sum it up, John?
Yes. Yeah.
So the next stage here would be finding out which of these combinations work the best. The way that we're going to know when we're done is really multifold. We're going to test the soil again. We're going to measure and see what's left in the soil. We're hopefully going to be able to do the same thing with the plant material, because some of what goes on is going on in the soil, in the biochemistry associated with the life that these organisms are bringing to the environment. And then some of it is uptaken into various parts of the plants. And one of the properties and reasons that poplars are so good is that they're really, really thirsty. They can take a lot of liquid into them. And so in a soil environment, if you're able to provide them with a soluble form of these toxins through the interactions of all of this biota working together, then the plants have the ability, especially poplars, to uptake and transpire a lot of that water, and at the same time drawing these toxins out of the soil, transforming some of it, and storing it, some of it, in their tissues. And this is the concept behind the whole thing.
And I will make a comment, if I may, Howard, that oftentimes we think in the field that this biological degradation mechanism driven by the trees works much better than it does in a pot study. And that's due to the sunlight and the scale at which the trees can then grow.
Yeah, absolutely. And expanding on what John is saying here is that, in the best of worlds, in the world of remediation, we're looking for results. And so the absolute perfect scenario for us to work in is in the soil, right in the area where the contaminants exist. And so in a perfect world, we would be doing this outside. Now, we're really fortunate here to be able to do this inside, and with a study of the duration that we have here, we should be able to tell within the 90 days of the study whether or not and how much and which combinations here are having effects on the contaminants themselves. But the proof really comes when you're doing it out in a natural environment and you can get this stuff growing outside. So I think, I feel like this is, I've covered this basically, although I know that it's complicated and new enough to a lot of people, so that I'm sure there's some things here that we can fill in and give you answer questions that expand upon our communication and how this stuff actually works.
So Howard, can you talk to what's the artificial light cycle that you're
Oh
going
yeah,
through today?
yeah.
And
This
how
is
much
through,
watering are you doing?
yeah, this is through John's advice. Although I found these lights, this is actually really great. I'm an amateur photographer, and I have all my great old camera equipment, including the light meters. And anyway, these are LED lights that are about $22 a piece from Harbor Freight, and they use an extremely small amount of electricity, but it's between the color temperature of about between 5,600 and 6,000, which is right in there for getting good vegetative growth. And if you look at the color of the leaves here as they're growing, they're nice and shiny and they're dark green, and so they're going to grow well. Now, the cycle that we have it on here is basically 17 hours on, and then the remainder of the day off. And so they're controlled by, you can look down here at here's the controller, a real simple digital controller that turns everything on and off. And so it kind of runs itself in here. And so what I do when I come in the morning is I find out with a temperature spin, and I have over here next to our controller, I have high-low recording thermometer, and I use added heat here. I have a small heater. It doesn't take much to heat this room. And I keep things right around 80 degrees Fahrenheit in here, which is a good temperature. And if you notice in the trees, we have all of these grasses that are starting to come in. And then there's some kind of a vetch I see. These are all from Willamette Cove. So we have a little bit of the cove growing right here in the lab. So the first thing I do is I look at how the plants are growing, and then I take a moisture meter, and I've recorded the starting moisture of all of these containers. And you know that the perfect, just kind of a rule of thumb for moisture is you really squeeze some hard, and if you can get just a little bit of a drop that you can see, that's about right. You want it pretty moist but not sodden. And so what I'm noticing here as I come in is that there are areas where the plants are using more moisture than other, and I think it has to do with the cycling of the air temperature around here, rather than the transpiration of the plants. Now, the placement of all of these plants here, it's done by a randomized design. I use a computer app that I plug in the number of replicates that I'm going to use on a given study, and I hit a number, or I hit a button, and it tells me where to put each of them. And so it's a truly randomized study. These things tend to work better the more replicates you have. Anyway, the way that the dice fell here is really pretty good because they're pretty evenly dispersed. So I look at moisture, and then I take pH measurements as well. And so I'm anticipating that what we're going to see, and you can chime in here if you have some knowledge of this, John, is it's been my experience that when I start a trial, a lot of times I see pretty low pH in the very beginning. And then I see over a number of days the pH coming up to more neutral. And then after a month or so, I see it starting to drop again. And I haven't done the chemistry myself, but I think what's happening here is that as the organisms are working on the organic compounds, they're putting organic acids into the soil, and that this, by measuring the soil pH, it's one way that we can track what's going on there, especially if you at the same time are able to do a little bit of analytical chemistry to prize out whether and where the source of these organic acids are coming from. And you know, we're a toxicology lab. We don't do that analytical chemistry here, but we can do simple measurements and then do the statistical analysis later, looking at the trends of the growth over time here. And so we record all of that, and then every week we'll have a report. We'll start putting these out. I have to tabulate the data that we've had so far. It's just been too busy here, really getting everything set up. But it's a simple process. And it's one that it's fascinating because the photographic record that I'm going to start putting up for everybody to look at, you're able to see every couple of days just a dramatic change in the amount of green material that's growing, as well as the fungi as it's starting to grow and starting to die off here and there on the surface of the soil. There's just a lot going on here. And, you know, at best, it would be nice to have a time lapse of the whole thing. And, you know, maybe I can do that at the end with the photos that I'm taking, but I'm not as sophisticated as I hope we'd be by now. But anyway, there's a lot of green here today that wasn't here a week ago.
So does anybody have any questions for either Howard or John you want to voice? John, maybe you can explain. Go ahead, Katie.
No, I just quick question. I just wondered if Howard, you could move the camera so we could see what the material from Willamette Cove was growing right now.
Yeah, you bet. Yeah.
Thank you.
Kind
of exciting.
Bare soil, Katie.
I think your finger's in the wrong place again, Howard.
That's right.
You're going to make a producer out of me.
There you go.
Yeah, here's the controls, and we should start seeing some green stuff growing here. I'm surprised that it hasn't.
That's what I was wondering about. Yeah, it's good to maybe get some pictures of that. I'm sure you're documenting.
Yeah. Oh, yeah, I have. I've been taking pictures every day of this. It's really interesting.
Thank you.
Yeah. And then here, you know, we have a little, it looks like some kind of a vetch or something that's growing there. Yeah, but a lot of green. And then here, it's just amazing watching this fungi grow. Now, the goal is not to grow mushrooms here when you're doing remediation. We don't want to create an attractive nuisance. And also that you know, most everything that's happening is happening below ground within the soil. And so in a real live remediation, our goal here is to be able to get a system working that will continue to work. And so it's been our experience that plant-based systems are something that you can just keep maintaining and maintaining. You can cut periodically, coppice the plants, and therefore remove and concentrate toxin in that way. And then I usually am shooting for a 3-year window in a bioremediation where most of what happens, it happens pretty quickly, usually within the first annual growth cycle. But sometimes, you know, it takes a couple of years just to get everything going. And so the goal really in bioremediation, and the thing that makes it attractive now, is that there is a restoration component to this, that by the end of the project, hopefully we'll be looking at clean soil and a clean environment in any of the projects that we do. Now, one of the things that I like to ask people when we give our talks on remediation is if any of you know what happened to the Alaska oil spill. Can anybody weigh in on this? Any of you know where the Alaska oil spill is at these days? It's got to be somebody.
We
Okay,
studied it from a natural resource damages perspective, but yeah, it's been what, 10 years, more than that?
It's been yeah, 20, over 20.
Wow.
But I meant physically where it is. It's in Oregon. It's in Oregon. And so this is what we're working to resolve, is so that we can use treatments like this that make it unnecessary to transport toxins and pile them up in another state.
So Howard, there was a couple of questions that I wanted to address. There was a question from Heidi about what kind of chemical analysis are we running and at what frequency. So I can answer that.
Go ahead.
We're running a full suite of chemical analysis. The baseline sampling we did was 2 samples, and really the analysis that we're running is one of the bigger costs for this effort. It's about $1,600 to do all of the analysis. So we're looking at the full suite of metals, PAHs, hydrocarbons, DOX and furans, which is a very expensive analysis, PCBs, maybe forgetting a couple, but we're running the full suite. Everything that we have at Willamette Cove, which is about everything, we're running for this. And the media that we're testing is at this point just the soil only. So at the conclusion of this test, we're running 3 strains, so we will likely do, well, with the control, we'll likely do 4 additional analysis at the end. And I know Howard mentioned that we would like to do an analysis of plant material. That wasn't in our budget at this point, but it would be nice to do that. But that's really answer to those questions.
Is anybody in the audience today from Robin Tanguay's laboratory?
Yeah, Robin was here earlier, and Diane Roman.
Are you still here, Robin?
No, she had to leave early. So
okay. Yeah, I have a question about that. That I spoke with one of the people there earlier about whether or not they were employing transcriptomic analysis, and this is something that I'm really hoping that we can do here, and I'd like for us to talk about that. And what this is, people, is that there's a number of ways that we look at toxins in the soil, and that we look at growing tissue and stuff like that. And one is through just DNA. And you have, and DNA is pretty straightforward. You are able to see the both strands of the DNA and get a signal and determine speciation and find out if the strains of bacteria and fungi and so forth that you end up with were the ones that you started with. But transcriptomics, it zooms in on this, and we're able to look at the transformation and appearance of compounds in soil and within tissues that are indicators of the production of organic acids and enzymes, for instance. And so it's a tool that we really are able to look at the mechanisms that are involved in the breakdown of these toxins, and to put a little clarity around that. It's felt that with fungi anyway, one of the main compounds is something called lacase, and lacase has the ability to break down polymers and the ability to degrade petroleum and so forth. And so that's just one compound. And with transcriptomics, we can look and we can see what's actually happening. And I think that Robin's lab is using this. It would be really, really something if we could have access to that at some point in the study.
I had mentioned before that we hope to do a virtual tour at Robin's lab as well to understand more what exactly her studies encompass. So we'll address that later. A couple of other questions for you, Howard. You stated that usually it takes a couple of years to get things up and running, but I thought I heard also that this test is 90 days.
No, most remediation, bioremediation, it starts happening right away. And so if you look at the production of enzymes and the breakdown of compounds, for instance, some of the first work that we did, I did when I was employed with Pacific Northwest National Laboratory. Well, you know him probably, Bob Wyatt, down at by the St. John's Bridge. We did a study there, and what you see is the relationship between the vigorous growth of these biological organisms correlates pretty directly with the breakdown of toxins. And so what we shoot for when we're doing this in a real live remediation environment, in the best of worlds, we get to choose when we do our planting, when we do our inoculations, and therefore we can make some expectations about when we're going to be done. And like, for instance, we've started with diesel contaminated soils within the Vados zone, which is the soil groundwater interface, and been able to completely remediate soils that were between 4,000 to 6,000 parts per million of diesel in the summertime. But you're not always that lucky, and that sometimes it takes a while. I say usually a year's growth cycle, so that you have the advantage of the benefits of the individual seasons and their effects on the growth of the plants and the fungi. And in the lab, we get to boost that time by turning the photo period up. So we're working on 17-hour days. So these organisms are working overtime right now.
Here's another great question that kind of gets to the point of why we have this integrated approach and how it works to treat soil contamination at depth. So the question is, at what depth will the soil be tested, and will different depth increments be measured? So you want to address that a little bit, Howard?
I think I'd like to turn this one over to John.
Okay, I was just looking at the plan here, and it looks like prior to the planting, the soil was homogenized, and they got the baseline soil data. After the test is concluded, it's my understanding that the trees and the roots will be carefully removed and the soil will be re-homogenized. And then that's how be sampled. Is that correct, Howard and Dwight?
Yeah.
Yeah, it's going to be the soil tested, and it will be basically that. We will take the remaining soil from each container that will be analyzed, homogenize it, and take a representative composite from that for analysis. The point I was hoping to have you guys talk about is how the hybrid poplars, the root system actually carries mycelia down to the depths of these roots, and
Yes, yeah.
it makes it effective at some pretty deep depths.
Yeah, we've traded soil at we've planted cuttings at 10-foot deep, and so it's just the soil groundwater interface is kind of everything. But the thing about the integrated biological approach is that the growth of the roots of the poplars are able to extend the growth range of the fungi in the soil, and they'll travel right along wherever a root goes. The mycelium can travel to. But
and when we talk
can
about
plant
this
it,
this mechanism, I'm sharing my screen. I'm hoping Dwight, you can see this diagram.
Yes.
So this is a diagram from Dr. Stephen Rock at US EPA, who's a phyto expert, along with James Landmeyer at USGS. It really shows the mechanism in general of how these organic chemicals are co-metabolized and sequestered and mineralized in the soil associated with the trees, how the rhizosphere degradation happens and is driven by the trees themselves through the photosynthesis and the transpiration stream, and also their root exudations. And so to answer your question, Dwight, indeed there are root associated effects, and as Howard mentioned, the roots feed the fungi, and additionally the uptake of some of these metabolites and breakdown products, and actually the parent compounds, happens into the trees where they are further degraded and accumulated. And this is the phytomechanism that's been developed by all the scientists and engineers doing this work at thousands of sites across the US to date.
Yeah, one of the great things that we're doing is that we're combining the use of fungi and the use of the poplars, so that we don't need to do a lot of excavation. We know what to expect in the root growth of the poplars, and so if we give a poplar, say, 7 feet in between plantings, we can do a core drill, plant the poplars, backfill with the soil from the cuttings from the hole, and 35 to 50% of the fungal treatment when we put the cuttings back in the soil, and you don't need to dig it up. As soon as things start growing and those roots move out there, the fungi moves with it. You don't end up having to tear up the ground, and you end up with the effects that you can see under the soil in the drawing that John put up there.
I see the next question was differential level of effectiveness at different soil concentrations of the contaminant. I think that's absolutely what happens depending on the phytotoxicity. I know there are hot spots that we think we can address, and I think that the site has the concentrations that are amenable to phytoremediation.
Yeah, when we test strains and get them evaluated for use in the environment, we go through a process where part of it is we subject them, like in the case of diesel, for instance, we'll actually grow them in the Petri dish on 0.5%, 1%, and 1.5% diesel, and look for the response. And as you look at it, it's really interesting because certain strains, they actually benefit from higher amounts of toxicity, and it just depends. But the suite of tests that we have that we go through, they include organic dye, colorization, and decolorization that have a correlation with the potential to break down hard to treat compounds like dioxins and PCBs. And so by the end of a trip that starts out in the woods and ends up in the lab here, by the time we're done with that, we have a really good idea of both the types of toxins that we can use these things on, as well as the kind of soil environments that we're going to grow them in. It's definitely not one size fits all. These are tailored systems.
Any other questions from anybody? Well, I think we'll wrap up. We're a little bit over. Thank you so much, Howard and John. Great tour. Very interesting stuff going on. Glad to see things are coming along so well, Howard.
Yeah. Yeah, and I'd like for all of you that as you puzzle over this, when you go home, if there's something that comes up, just write us a note, and get around to it and respond for sure. I'm happy to do that. And you know, this is really I'm enjoying the study and already learning things and seeing unexpected things happening here. So it's going to be fun to watch.
Thank you, Howard. Dwight, when will the recording be available?
I'll have to figure out how that works. I know we're recording. We got an hour and 10 minutes, so we will have a link, I assume, available that we can send out.
Yeah, great. Hey, thank you.
Thank you all so much.
Yeah, thank you.
All right, thanks, you guys.
See you soon.
Take care. Have a great weekend.
Bye-bye. Yeah.